Okay, gang, here's your video on section 2 of chapter 11. This is specifically about the reception of a signal, and the first picture I have up here is the picture that I ended the last video with. It just talks about when we're communicating, two cells communicating. Typically on the receiving the signal cell, there's three steps that are involved in a cell receiving a signal and then actually doing something with the signal that they have received. So there's the reception. First step is the reception of the signal. We're receiving the signal. The second one is transduction, and then the third step is activation. So this entire video here is focused on the receiving of the signal, or the reception stage here. So first of all, we can take that and fit it in the whole receiving or reception, have that fit into two different classes. The first would be types that are signals that are received inside the cell, or intracellular receptions, and then the second type are ones that are on the surface of the cell or in the membranes of the cell, which we'll get to in the next slide here. But intracellular reception, the example they use here is testosterone. And first of all, the definition of intracellular reception, all you need to know there is that the signal is received by a receptor protein inside of the cell. Okay. Now, specifically the example that they're giving, if you remember, I had said that all these types of communication are going to be different with every single communication signal that is sent from one cell to another. It's always going to be responded to differently for every different scenario. So this happens to be the one that they're choosing to focus in on for the intracellular reception, and it is dealing with testosterone, which I talked about in the last video. But testosterone is a very hydrophobic molecule, and so it actually passes through cell membranes really easily, if you remember those characteristics of cell membranes. But so small particles can do this as well. But testosterone and a lot of other hormones actually are able to just pass directly through cell membranes, and in doing so they can enter any cell in the body. When they get in there, if it's a cell that happens to have a very specific receptor protein that recognizes testosterone, it will link to that, and in doing so it just activated that receptor protein, and then action can happen. In the case of testosterone, what it does, which is a really unique thing, but when the receptor protein and the testosterone join, they actually go into the nucleus of the cell, and they start to act as something called a transcription factor. Anyway, a transcription factor basically turns on a section of DNA and says, hey, use me to do something, okay, to make a protein, which is what DNA does. And so this will actually go and link up to a section of DNA and basically turn on a gene. And turning on that gene, the whole process of making a protein happens. And so in the case of testosterone, these genes that they turn on are the genes that lead to male characteristics, male sex characteristics. Anyway, and that's through that system. So that's intracellular, simply because this molecule actually attaches to a receptor protein that's already inside of a cell. And this one ends up being a very simple process. Testosterone and how it works is a very simple process. Wanted to introduce a word that the book, as I was looking through here, the book says that it doesn't really define well, and I don't know that it's shown up at all this year. There's a lot of words that haven't shown up at all this year, but anyway, it's ligand, L-I-G-A-N-D, ligand, and basically, I don't know why they decided to throw a different term in here, but a ligand is basically a substrate, okay, on an enzyme. A similar idea. A ligand is something that very specifically is recognized by a protein and fits into basically the active site of a protein. The reason it's not, I do know the reason they don't call it a substrate, is in this case it's not actually attaching to an enzyme, it's attaching to a protein, and so they don't call it a substrate unless it's actually an enzyme that it's attaching to. But it's the same concept. We would call it a substrate, or a ligand is what you're going to see several times in the book. So, alright, we'll get on to the receptors that are stuck on the surface of a cell next.